final speaker for today is Dr. Dean Jones from Emory University, and Dean is going to bring us back to the most important part of risk assessment, which is exposure assessment.
Thank you, Yvonne. It's a pleasure to be here, and hopefully you haven't all exceeded your thresholds for the day. Although, for Maurice, I'm not sure I'm going to be able to impart too much knowledge, but maybe some data. I'm going to talk today about our work on what, using the term here today, universal biomonitoring. And I'm in a Department of Medicine, actually in a pulmonary division, although have not studied very much in the way of pulmonary disease. But what the reason I moved from biochemistry department into Department of Medicine was really to address the problems of personalized medicine. And what the starting point really on this is that when we think about healthcare, we really have two main drivers that begin with the letter C. One of them is complexity, that there is such a tremendous complexity in disease that it's really very difficult to, if we try to think about personalized medicine, to be able to address any individual disease on a personalized basis without considering the comorbidities of the individual. And this means that by necessity, everything comes into, we have to deal with complexity. There are many aspects of that in terms of the diet, the microbiome, and importantly, exposures that have to be addressed. Now, the big problem when we come to health care in terms of drivers is the other C, and that's cost. Because the cost of tests is considerable. And if we begin to think about personalized medicine and trying to think about the spectrum of disease processes that we have to address, we get to the point where we really have to have hundreds, if not thousands, of tests in order to actually cover the broad spectrum of different risks. And so what one can see is that if we really wanted to do this with the current paradigms of the way we measure things, the diagnostic tests we use, we would simply not, none of us would be able to afford to even have an annual physical examination. So when I moved into the Department of Medicine, the main motivation then was to try to come up with a different strategy for addressing that aspect of diagnostics. And I did this comparison based upon the really as a basis of just constantly hearing the genomics folks talk about the cost was going, how the cost was going down because the speed of gene sequencing was increasing, and every year or two you would hear talk about the doubling of the ability to sequence. And so I went back to think about the issue in terms of diagnostic testing and analytic biochemistry, if you will, and just thought back that in 1957, Moore and Stein introduced the automated amino acid analysis. Quite a remarkable feat to be able to, in basically about 6 hours, be able to measure between 20 and 30 chemicals in an automated sense. This was truly a breakthrough in terms of analytical sciences. If we fast forward a half of a century, really the best that was available in 2007 was about 300 analytes that could be measured. Metabolites, if you will. But this was really with a combination of techniques, and if you thought about the cost of that, well, wasn't outrageous, but it was still in the hundreds of dollars, and if you talked about the time frame, it was in the period of, if you send it off to a commercial lab, it would take a month to 6 weeks to get your data back. So one could argue that in a half of a century, analytical methods really had not improved very much as far as measuring metabolism. So we focused on ways, tried to go through different possibilities for how we could really make a dramatic improvement in that. And if you look on this on the right here, see is an illustration. I don't have time to go through the different techniques, but the high-resolution mass spectrometry was introduced in 1974 by Alan Marshall and Comesso, was a collaborator on that, introduced the Fourier transform ion cyclotron resonance mass spectrometry. And that method really had not been applied to in any major way to analyses of metabolites. And so what we decided to do was to try to apply that method, and the goal, and we received funding from the National Institute of Environmental Health Sciences for this, was to try over a 5-year period to go from that ability of being able to measure about 300 analytes to being able to measure about 1,500 metabolites. And we were specifically focusing on plasma, but this also works for urine and other biological materials. Now, what was really quite unexpected was that already within the first year of the grant funding, my collaborator on this project, Qianwei Yu, had been able to write. He's a computational biologist, and he was able to write new adaptive processing software to be able to extract information better. And so already we had fulfilled the goal in 2009 of being able to measure 1,500 metabolites in plasma. In 2010, Quinlan Salto was a postdoc in the group, applied a dual chromatography method and more than doubled that number. So we were now up to over 3,000 metabolites, and actually up to 7,000 in some cases. Then we did something that was really, I felt essential to be able to get to the problem of human exposures, and that was that we were doing all of our work in a shared instrumentation facility in the chemistry department, and we were finding that occasionally we would get fingerprints of chemicals that were actually chemicals that that chemists had been studying in the facility. And so we decided that if we were going to look at human exposures, that we were really going to have to change facilities. So we obtained another high-resolution instrument, which we moved into my lab, and quite remarkably, the ability to detect analytes in human plasma increased more than fourfold, closer to fivefold. And so in the past year, we've a very talented graduate student, Karan Uppal, has written a new series of algorithms called XMS Analyzer, which again enhances the data extraction capability. So now we're able to capture in a single 10-minute analysis over 45,000 ions. Now, that's not exactly equivalent to metabolites, but these are essentially, each of these are, or almost all of them are, in fact, isolatable individual chemicals that are in ionic form. And there's some redundancy in that, but by doing the dual chromatography method, we actually have a total of about 95,000 ions. So what it really does is it puts us into a realm of being able to get more or less a universal type of sampling of chemicals that are in biological samples. The other thing that it does is that was really very important from Karan's method is that it allows us to be able to look at data according to the quality of the data. So this is a plot of the coefficients of variation of the analytes, and with the you really can set your software to be as rigorous as you want. And in this case, we have it with an average coefficient of variation of a little bit below 30%, which is actually pretty good for most mass spectrometry methods. what you can see is that there's over 2,000 metabolites that we're measuring here that have coefficients of variation of less than 10%. So it's possible to have really very good quality of the data. And so the way we do this is we run every sample in triplicate. That allows us to be able to get to really know within an individual whether an unknown chemical or an ion that we detect only in that individual is actually reproducible in that individual. So there are other aspects to this that I don't have time to go into, but I think the point is that we're now at a point in terms of the methods development that it is feasible to be thinking in terms of the concept of more universal coverage. Now, you've already seen the KEGG pathway. This is, if you look at the pathway, there's a bit over 2,000 metabolites in the chart. In one analysis, we will get over half of the metabolites in the chart, at least in terms of the high-resolution matches. There's some ambiguity in this. Obviously, it's very difficult to go back and confirm the identities of all of the chemicals. But in a pathway sense, one can, in fact, even if one of the chemicals is wrong in a pathway, when you have 4 out of 9 chemicals that you're measuring, you can get pretty good estimate of the pathways that you are correctly measuring. So at this point, we have a platform that is, we run this in a very routine manner. That's really one of the keys to making it work. It runs 24 hours a day, 7 days a week. Once we start an analysis sequence, we run it for typically over a month, and then we have very strict standard operating procedures. We know how long the columns will last. We know when we have to switch things out. We don't do the obvious thing that every analytical scientist does, and that is put on reference standards, because in fact, when you put on a reference standard, then you never really know whether anything subsequent that you measure is a consequence of what you have put, the standards that you have put on. The other problem, of course, is that your standards are never pure. And so when you start putting additional things into the system, you begin to put risk and variation into your analytic platform. So the real key, I think, to being able to do this in getting good data is actually doing it in a very routine way, where one is measuring, for instance, a series of urinary samples, only urinary samples. If it's plasma, only plasma samples, and doing it in a very continuous way so you don't have interruptions of column flow and things that are going to distort the analytic capabilities. So it's very useful in the terms of ability to pick up pathways that are being impacted. This is a study that Quinlan Salto did so this is unpublished data, but this is on dialysis patients where she's been looking at frailty as a characteristic. And so looking at the metabolites that are associated with frailty, what you can see is that indeed there are regions of the metabolic chart that are enriched in metabolites associated with frailty. So I think it is going to be very useful in terms of monitoring biological effects. Another example I just wanted to show quickly is specifically a study of exposures and Parkinson's disease. This was a study with Beata Ritz, who's at UCLA, where she has been collecting samples from patients with Parkinson's disease and has them categorized according to occupational exposures to pesticides and also progression of disease. I'm not going to show you much of the data other than to point out that if one looks at the metabolites that are really associated with exposures, what one sees is that there are many metabolites that significantly differ. If you apply pathway analysis to this, you can, in fact, identify specific pathways that are impacted. So, again, this is very early in terms of development, but I think that it's an approach that is actually going to be very useful to be able to define risks of exposure and also give information on pathways that are impacted. So, summarizing these points, we can pick up metabolites in 146 out of 154 pathways. We can identify, at least in model systems, we can identify pathways that are perturbed by chemicals, and I think this is going to be very useful in terms of linking exposures to mechanisms. Now, we've done a study which was actually difficult to get published because it's a little bit, it's difficult to explain in any circumstance, but we have really a massive amount of data that we're getting, and we wanted to ask the question, can we use that to sort of test different animal species and really see how different humans are? And our hypothesis was that humans have such a varied exposure that humans were going to have a lot more environmental exposures than laboratory animals. So we took plasma samples from 7 different species, including humans, and 2 other non-human primate species, and then examined them with regard to their metabolic characteristics and asked the question, which metabolites vary more between species than within species, and which metabolites have pretty much the same or have no difference as far as the interspecies variation than they have intraspecies variation. And the data turned out to show 2 modules, what we call here Module 1 and Module 2, and these are the metabolites that basically vary the same, have the same type of characteristics regardless of species, interspecies comparison, whereas the Module 2 are ones that are really more species specific. Now, what was most interesting about this from my standpoint was that when we looked at what the chemicals were in Module 1, these are mostly intermediary metabolites, whereas the chemicals that we had detected as environmental chemicals were essentially all in Module 2. And so I think the take-home from that is something that we've already heard a bit about today and that everyone's aware of. It is the problem of extrapolation across species, that in fact the different mammalian species do have great differences in terms of how they metabolize foreign compounds, environmental compounds, and the impacts of those. We've gone through and we've done MSMS on these, and now I don't know how many we've done, but a large number, certainly not going up to that 50,000 number that we're detecting. But the point is that there are a lot of environmental chemicals that are found, and they are found in the different species, regardless of whether they're eating, regardless of humans versus the animals living in captivity and in our best husbandry conditions. It really is just saying that the water supply and the food supply is probably the major source of the contaminants, and I think that's what everyone would accept anyway. So I want to now talk about what I think is really the potential important application of this technology, and that's based upon this study by Patel and others on the environment-wide association study that they did now a couple of years ago, looking at the type 2 diabetes risk and association with specific exposures in the NHANES data set. This is a Manhattan plot, basically the same type of plot that's used for GWAS studies. Now, we've done a study which was actually difficult to get published because it's a little bit it's difficult to explain in any circumstance, but we have really a massive amount of data that we're getting, and we wanted to ask the question, can we use that to sort of test different animal species and really see how different humans are? And our hypothesis was that humans have such a varied exposure that humans were going to have a lot more environmental exposures than laboratory animals. So we took plasma samples from 7 different species, including humans, and 2 other non-human primate species, and then examined them with regard to their metabolic characteristics and asked the question, which metabolites vary more between species than within species, and which metabolites have pretty much the same or have no difference as far as the interspecies variation than they have intraspecies variation. And the data turned out to show 2 modules, what we call here Module 1 and Module 2. And these are the metabolites that basically vary the same, have the same type of characteristics regardless of species, interspecies comparison, whereas the Module 2 are ones that are really more species specific. Now, what was most interesting about this from my standpoint was that when we looked at what the chemicals were in Module 1, these are mostly intermediary metabolites, whereas the chemicals that we had detected as environmental chemicals were essentially all in Module 2. And so I think the take-home from that is something that we've already heard a bit about today and that everyone's aware of. It is the problem of extrapolation across species, that in fact the different mammalian species do have very great differences in terms of how they metabolize foreign compounds, environmental compounds, and the impacts of those. We've gone through and we've done MS on these, and now I don't know how many we've done, but a large number. Certainly not going up to that 50,000 number that we're detecting. But the point is that there are a lot of environmental chemicals that are found, and they are found in the different species, regardless of whether they're eating, regardless of humans versus the animals living in captivity and in our best husbandry conditions. It really is just saying that the water supply and the food supply is probably the major source of the contaminants, and I think that's what everyone would accept anyway. So I want to now talk about what I think is really the potential important application of this technology, and that's based upon this study by Patel and others on the environment-wide association study that they did now a couple of years ago, looking at the type 2 diabetes risk and association with specific exposures in the NHANES data set. This is a Manhattan plot, basically the same type of plot that's used for GWAS studies, and the idea is that you plot the negative log of the probability, the p-value here, and then that allows you to have a significance line, and it tells you which chemicals are potentially associated with that risk. And you can really do this with any disease risk factor or any population that you have, and you can set this up. It's quite a versatile way to look at the data. So we've done this on a few studies now. I want to just show you a couple of examples. This is what we call a metabolome-wide association study, and I'll get back to why we don't use the term environment-wide association study, but I think the MWAS is a more appropriate term here. What you can see, this is a study with Daniel Promislo, who's a Drosophila geneticist, and we've been studying, we have a grant from National Institute of Aging on the metabolomics of aging. And Daniel has been looking at the at Drosophila and the aging and metabolomics of aging in Drosophila. And what you can see from this plot of metabolomic data with, again, a Manhattan plot is there are many different chemicals that are associated with aging. And the nice thing about this type of an analysis is you can see both positive and negative associations with the aging. You can also put in different criteria for the quality of the data because we're measuring everything in triplicate, and then can also see the number of non-significant features. Many different important findings from this, but I think the most important one for today's topics are really that these are laboratory flies. So these are flies that are in our best husbandry conditions that we can have for a fruit fly. And I guess quite unexpected from my standpoint was that some of those dots up there are actually pesticides. And so the implications of that to me are pretty big, because it's saying that even with the purified diet that there's enough pesticide residues in the fly food that it's impacting the fly longevity. I don't know what the implication of that is with regard to what we're eating, but I think it's something that it tells us that this type of an approach can potentially be very informative as far as the exposures and risks. We've also done this an analysis in terms of the metabolomic associations with the SNP variation, so we can do basically a gene metabolome interaction study, and you can see there's I guess this is pretty much cut off here at the significance level, but you're seeing a very large number of metabolites that are significantly associated with variations in human population. These are healthy people, work done with Greg Gibson, collaborator at Georgia Tech. We can also look in terms of other diseases. This is a smaller study, really pilot in nature, but it shows again that there are many metabolites associated with Huntington's disease. We've done other studies on, this is the metabolites in severe asthmatic children before being treated with corticosteroids, and then being analyzed after being treated with corticosteroids in terms of responsiveness. And so basically this is the metabolic profile that's associated with either resistance or sensitivity to corticosteroid therapy. So again, in the context of today's discussion, I think we really, in a certain way, can turn risk assessment upside down. We can now look at populations, and instead of making a presumption as far as what chemicals are potentially causing risk, we can ask the question, what chemicals are associated with risk? And then we can go back in mechanistically and test those hypotheses. So it doesn't really, it's an association, doesn't prove anything by this type of an analysis, but from the standpoint of discovery, it can tell us in human populations which of the chemicals are actually associated with whatever risk factor we want to look at. So I wanted to come back now just a couple minutes on this concept of why I think we should be calling it an MWAS, metabolome-wide association study, rather than an exposure or environment-wide association study. And that has to do with taking a look at the pan-metabolome, that is the total picture of what we see. I showed you already the KEGG pathway, which is really those 2,000 metabolites or so that are considered the metabolites that are formed based upon our enzymes within our body. So we call that the core nutritional metabolome. There's a very large group of chemicals that we get from our diet that are really non-nutritive in nature, and we call this, combined with the others, the food metabolome. Estimates are that there's about 2,000 different chemicals that we typically get from the plant, from plant food sources. Let's see. Decided I've talked enough because it's not letting me advance. There we go. All right. The next group of chemicals I think we really need to pay attention to, it's already been mentioned today, is the microbiome-related chemicals. This is really largely uncharacterized, and estimates are that it's perhaps 10 to 40% of all the metabolites in our plasma are actually derived from the, or in some way impacted by the microbiome. There's greater than 1,000 pharmaceutical agents that are in use, so we have a good number of chemicals that are going to be derived, and in fact we see these in our samples. Also a large number of chemicals that are derived from the various nutritional supplements that are used. And then we really get to our environmental metabolome. So I think that in terms of the variation and what we see, and certainly in terms of health risks, we can't really ignore all the rest. We have to consider the exposures in the context of all the rest. And so even though there's a very large variety of these agents, I think that they are oftentimes lower in abundance, but they are impacted by the microbiome, and definitely in terms of their human health impact, their health impacts are really going to be dependent upon the other components, the drugs and the nutrients that we consume. So again, I think there's real potential for the use of this metabolome-wide association study and the gene by metabolome and epigenome by metabolome studies as far as moving forward and really being able to understand and I think better predict risks. And I think this approach is really where we have to go in terms of operationalizing the exposome. The concept that we can now do a cross-section of exposures from a metabolome-wide study, if we had this type of information across the life cycle, then we really have the possibility of being able to get that operationalizing the exposome, that wonderful concept that many of us have been so interested in the last couple of years. So linking this back to utility in terms of mechanism, we've done some just pilot studies where we've done metabolome by transcriptome studies. These are in vitro studies. What one can see is that there really is a very nice concordance of metabolomic signatures and transcriptome signatures. And so I think that one could combine transcriptome studies in human populations with metabolome studies and actually have a way to really get information on the mechanisms. So this, I believe, will help us in many ways in terms of things that are really very difficult with regard to the way business has been done based upon targeted analysis, because we have such a broad spectrum of analyses. By using one platform that gives lots of information, we can overcome this problem that targeted analysis is very expensive. We also can overcome the problem that targeted analysis is based upon toxicology as our starting point, because now we can actually look at what we're actually exposed to, or at least get some better perspective on that. It also allows the epidemiology to be done really, again, on the biomonitoring of the chemicals that are within us rather than only on a limited subset. And so I think in that, plus the issue of the complexity is really very important, because now with this approach we can actually ask do the biostatistics to evaluate the mixture problem. So just now finishing up, so stop just for a second, take a breath on this one. I want you all to look at my number up there at the top, and I don't think this is a pipe dream. I think given that basically in a 4-year period we've gotten up to being able to measure over 50,000 ions in 20 microliters of plasma, I think with some clever analytical scientists who really understand the concepts of chemical space and application of techniques that are available today, I really think that we probably could have a platform where we are delivering 500,000 chemicals. We actually could do that in a cost-effective way. It costs about $125 per sample, but we do that 3 replicates, and we do it on 2 columns. And so it's not a stretch of the imagination that we could really do this by incorporating it into human health care, that we could really get this type of information on hundreds of thousands to millions of people, that we could in fact be doing this over the lifespan of individuals. And so I think that this really puts us in a place where we can really be thinking about risk assessment in a very different way. So I mentioned we could do this if we could actually incorporate it into annual physicals. There are many other ways that we could do this. We could do it on archival samples to really get a head start on things, because there are many archival sample sets with really good information on them. We could incorporate it into newborn screening. If we wanted to worry about all of the HIPAA problems, we could actually do it in the veterinary clinics, because so many of our pets actually eat the same food we do and live in the same environment. So to summarize, enhancing systems biology. I didn't talk about systems biology at all. We try to think about it, but it's actually, as everyone knows, it's very difficult when you start trying to apply it to a human population. But the charge is to think about this in terms of environmental exposure. I think that the universal biomonitoring gives us a good place to start. Maurice, you can see why I believe in the data. I definitely think that we need to go develop the models, but then ultimately back to the human and the individual, we will need the data on the individuals to be able to make those good predictions on a personalized basis. I think we'll want to go with this MWAS concept and the gene metabolome interactions, that there's a lot of power there. And ultimately, I think that's where we want to go, is to be able to take this to the individual level and hence be able to do more than just a population-based risk assessment, actually do a personalized risk assessment. I think ultimately that's going to liberate a lot of chemicals that currently are not used, specifically a lot of therapeutics that are not used, because if we can predict who's at risk of what, we can flip things around and actually introduce chemicals that are currently not used because of the problem of risks in some populations. Lots of people involved in this, and there we get the last slide. Most of the data that I showed was done by Youngsha Park, Quinlan Salto, Sky Lee, Jennifer Johnson, and Karan Uppal. And over there on the bioinformatics side, obviously everything that I talked about would not be done without the very talented, capable bioinformatics collaborators. So thank you.
Thank you, Dr. Jones, for a very provocative and informative talk. I just wanted to ask a couple of clarifying questions. When you're talking about 500,000 chemicals, can you be more specific about which of these molecules essentially are endogenous or nutrition-derived molecules, and how many of them you would classify as environmental chemicals?
Unfortunately, at this point, we really don't know. But I would think that by 2 different methods, we're coming up with in the range of 1,000 to 1,200 chemicals in the plasma. When we do cell extracts, we get a different spectrum of those, but it's again in that range of 1,200 or so.
And you're talking about environmental chemicals rather than biochemicals?
These are biochemicals. These are endogenous. So I'm talking about out of that 50,000, we will get... if you start looking at how frequently those are found in people, Things such as we'll pick up some of the synthetic juvenile hormones that are used in flea and tick collars. We'll get those only in a certain fraction of the people. If you look, certain of the chemicals, for instance, there's some of the chemicals that are in hops that are used in beer. You're going to see those only in some people. Resveratrol metabolites you're going to see only in some people. Rotenone, for instance, you don't see in very many people. You see in some. Rotenone, there's a rotenone contaminant that's in virtually everybody. Perimicarb, everybody.
And on the, going from, even very precise mass to the actual name of the metabolite, this is a probabilistic, as you know, you get a list of things that could be that. So how high is your confidence in actually naming names and, 50,000 peaks that you're seeing?
Oh, well, we don't have the number, we don't have the staff of chemists or facilities really do much in that way. Right. When we've looked at metabolites that are in the metabolic chart, it's between 60 and 80% of those are correctly identified because we can do MSMS on them. We put the authentic standards. We haven't gone very far on that list. So, but for those for that first 1,200, it's pretty secure. The others. What's interesting is you have such high mass accuracy, you basically can get the elemental composition. And so in the metabolic list that you don't have very many matches. Actually, over half of the ions that we pick up, we have no matches. So there's half of those are completely unidentified. It doesn't mean that they're not known chemicals. It just means that we haven't identified. So we picked out some, such as the rotenone, perimicarb, the ones I showed up there. We did the MSMS on them. We pick up a lot of flame retardants. We've verified identities on some of them.
Would you venture a number of actual chemicals that you can measure? I mean, I'm trying to get at whether you're now a real competition for NHANES with their 500 environmental chemicals or not.
I don't think there's any question that we could pick up in one analysis that we could pick up hundreds because we pick up hundreds of chlorinated chemicals that are.
And you can get a concentration as well, right?
You can't. In this protocol, the whole problem of quantification, with absolute quantification, is you have to have the standard.
Right.
And so when you start going up to 1,000 chemicals, you're talking about well over $1 million to get the standards if they were available. And so that's a limitation. And I think the point is you can go back, we validated this, you can go back on any one of those and determine its absolute concentration using those methods. But as far as the overall, it's going to have to be a computational approach where we will be estimating the concentrations, not by authentic standards.
Please, let's start with the microphones.
Thank you very much. That's really very enlightening talk. And I'm Suri Wulimuri from EPA. I have a couple of questions. One thing is, I think from the hazard identification point of view, metabolomics has a lot of promise in terms of identifying the metabolites. And the other aspect is dose response. Like with your studies, do you see any dose response for the metabolites? Like, have you done any? I mean, I do understand you need to have a standard for that and then for the quantitation.
So we have done very limited work on that. Certainly in the model systems where we apply the metabolomics, you do see a dose response. We've done a little bit of nutrition related work in humans where we see dose response. So I think it's clearly there. It's going to require, we've only really had this up and running in a routine way for less than a year now. So it's going to take a good number of studies and probably a good number of laboratories in studying this type of an approach to really get at that handle.
Yeah, the other question I have is regarding the internal standard. You mentioned that you don't use the internal standard because of any interference or misjudgment for metabolite. Is that right?
So we have two instruments. I guess I misled on that. We have two instruments. One that we use in the chemistry department, we use standards on it.
Okay.
The other instrument we have in my lab, we don't put anything on it. And we do have, in every sample gets a mix of internal standards. That's constant for, we have a mixture of 14 stable isotopic standards that we put in everything. And then every 21st sample is a mixed reference sample. So we've built in a structure so we can get a relative quantification down the road. We, in principle, can actually estimate concentrations of anything down the road. But what I meant was we don't come back on a routine basis and put junk, I mean, I'm sorry to call standards junk, to put standards on the column.
Yeah.
That's all I meant.
Okay. And you mentioned there are like 50,000 ions you're able to detect.
Yeah.
So, but the metabolites you can detect is only for those which have the standards, right?
Well, no, because this is the instruments, we use an Orbitrap, which is a hybrid instrument, and the other one is the LTQ-FT, which is also a hybrid instrument. And those have the capability of doing MS on them. And so what we do is we run the routine analyses, just running them, but then with each sample set we will run an MS scan, so we get the most abundant out of the 1,000 scans or so we do in an analytical run. We get the 5 most abundant ions that we fragmented. So we actually have that information, but it's very labor intensive to go back and confirm the identification. So that's why we do this inverted approach, and that is you pick up what turns out by the MWAS to be associated, and you only target those for identification, rather than try to identify everything.
Let me ask you a question from one of the online questions. Have you tried longitudinal studies on same individuals to try, time of day and some other things? And then you did mention what is the volume, which is 20 microliters of serum that you're using. But what is the processing of that blood? How quickly that can be, should be processed for it to be useful.
So there's 2 papers on this. One was in the Analyst in 2010. Jennifer Johnson was the first author giving details, and the second one was Quinlan Salto that was published last year in Metabolomics in the journal. Basically, we tried to develop this for clinical use, and so the concept was that we would use just a standard EDTA plasma, so nothing special as far as the collection, and we would simply do a protein crash as far as the processing. So we typically use acetonitrile, but 2-to-1 with the sample, but we've also done methanol, and both work very efficiently. So it's very simple processing. The concept of stability, some things are going to be unstable, but from this type of an approach, you have to assume that if it's very unstable, it will break down to a stable product, which will still be a useful biomarker.
And the longitudinal or different times of day, because I think there's some data coming out of the NHANES suggesting that the actual levels of chemicals that they report are quite variable, even on this, during the same day.
So it's we've done one series of studies where we did 34 samples of the same individuals over a 10-day period. If you just do a principal component analysis, the samples for all of the subjects except for one, all of the samples clustered together. So the overall character of the metabolome is truly individual, but there are clearly changes as far as individual metabolites during a 24-hour period.
Richard?
Richard Dennison, EDF. Very interesting talk. Thank you. You mentioned population-wide and then individual, and I wanted to focus in between those at a sort of subpopulation level. And you did describe how you've looked at the differential patterns you see between severe asthmatics and normal individuals, for example. But I'm wondering if you or others have already applied this, or if there's any reason why it could not be applied in the very near term to looking at differential populations based on exposure differentials. So, for example, rural versus urban dwellers, or people based on proximity to roads, looking at potential air pollutant effects, et cetera, et cetera.
So we have some projects underway on that. The one project with Biarritz was actually an exposed population. That showed much stronger effects of discriminating rapid progressors from slow progressors as far as Parkinson's disease rather than an exposure. But the samples were taken very long after exposure, so you wouldn't expect for any of those direct metabolites or the environmental chemicals to actually still be in those samples. So clearly those are types of studies that we need to do. This is, like I say, it's relatively early on. We've been, up until this year, the largest study that we had done was 100 individuals. We've now broken the 600 barrier. And so the concept is we're going to, hopefully over the next year, we're going to be measuring between 5,000 and 8,000 samples. So we're really going to have a lot more of a reference against which to compare things, and then we'll be able to see, well, I'd be able to answer a lot of questions with regard to subsets and how they really map out in the overall range of samples that we pick up. But clearly, those are the types of things we want to do, and any of you who have good sample sets, we're definitely open to conversation on getting them run, because, like I say, we're at a point now where I think we have a stable platform, and we're collecting the data in a way that can be a cumulative database. So, and my goal is to make all of this open access, because there's too much information here for us to actually get all of it out. We want to make this so that people can mine the data and answer very specific questions.
Thank you.
Let me just follow up quickly. So when you would release this data, what type of identifiers you would release? Just mass charge ratio, or?
Well, so what we've published so far, we have it has really been the accurate mass, the retention time on the column, and then the signal amplitude, the integral. Obviously, to make it really useful, we're going to have to put some types of at least demographic information, but we have to really be careful on how that's done to avoid identifiability.
I also would think that to make it useful, you need to, maybe make some identifications of your peaks or ions. So have you done that?
Yeah. So we've done that. We have a master list of identifications ones that we have identified so far, but even those have different quality of identification, because some of them we've done with authentic standards fragmenting directly. Others we've identified them based upon the fragmentation pattern compared back to the NIST samples, the NIST data.
And how long that list is, and from your real high confidence, then how many of those, or percentage-wise, would be biochemicals or natural metabolites versus environmental chemicals?
At this point, almost everything that we've done has been natural metabolites. We have not extended it very far. We're getting the list of chemicals. Ray Tisus sent us the lists of chemicals that they have, their standards, and so we will be running some of those to try to match them up. But we're still personnel limited on being able to do a lot of that.
Next question.
Don Madison, Risk Sciences, was really interested in your pediatric sensitive and resistant asthma population, and it suggests a couple of questions around stability of disease signatures and the persistence of disease signatures in the presence of treatment.
So, I guess you want me to comment on that. We haven't.
No. It was just.
It does. I mean, you're absolutely right, and we haven't done a whole lot on that. We have done one study in intensive care unit patients where we have an intervention where, some of the patients get better, and we have, and the intervention's actually beneficial, and one does see in that type of a study, if you do principal component analysis, the intensive care unit patients really differ from normal people, and the ones who recover, they have a trajectory back towards normal. And so I think there are bioinformatic ways to really address that and then go in and look at specifically what metabolites are really the predictors of recovery. Again, we're very early on those applications, and hopefully there will be other people who pick this up and actually can run with it, because that's going to be very powerful.
Yeah, hi, Jerry Poggi, SOEH. Great talk. You're kind of projecting a rapid change in technological advancement. I mean, even your techniques and analyses just seem to have been increasing at a geometric pace. I wonder if you would care to speculate, here we are 5 years into toxicity testing in the 21st century, where you think we would be 5 years from now in terms of numbers of laboratories, the number of data sets, the ability to share information, and to get to some better interaction with P4 medicine using environmental exposures as one important component.
Well, I've done a lot of thinking on that, because the potential for medicine, I think, is so great. And I think one of the things that I've learned from really being more active in the medical research community is that in my pulmonary division, they don't think about the environmental factors that actually could be contributing to the pulmonary fibrosis or the other risk factors. That's not in their radar. They don't think about that. Cardiovascular disease, even though we know particulates affect cardiovascular disease, it's not in the thought process of the clinical cardiologists, even the very best of the research group. So as I've been thinking about this, I think that there's an absolute need to try to move this as rapidly as possible on the clinical side, because I think it's going to be very informative. Trying to convince them that this should be done is it's a big challenge. But I think from an environmental side of things and environmental risk, that it's actually much more tractable. Because if we can get this standardized, so my goal is to do this type of an analysis on 100,000 people, where we have, within that 100,000, we have people with known exposures, where we really can classify them and can compare what their metabolic profiles are to all the other people in that analysis. That can be done. I mean, over a 5-year period, if we had 2 more instruments, we could do 100,000 samples. So that's not out of the range of possibility at all. It has hurdles because of the issues of HIPAA, which means it has to be structured in such a way that it's truly de-identified, but yet has enough information associated with it that it's going to be useful for data mining. And so I think it can happen. It can happen in a fast way. Whether it will, there's obviously hurdles, and those are the ones that we're all just going to have to address, because if we can't get past those types of barriers, we're not going to be able to get there.
Thank you very much again.